It's not. Yeah, so now we are connected. So hello everybody and welcome to the third seminar on the 10x genomics and single cell technology. So I'm going to start in a couple of minutes so everybody joins, so they will have a little bit more time for everybody to join. It's 10 o'clock, so we can wait a couple of minutes. People is connecting. So just for to start with, just saying that today's seminar is the topic is oncology and cancer biology, and we have today Dan Carter from the School of Biomedical Engineering, and also has a conjoint appointment with or joint appointment with the Children's Cancer Institute at UNSW. So he's going to be the invited speaker for today. And as usual, we're going to start with Yvonne Peterman, which she is the, today I'm going to say your title properly, is the Science and Technology Advisor for 10x. And then she's going to tell us a little bit on the 10x, I guess, portfolio and solutions, and highlight a few of the applications that 10x can offer for oncology and cancer biology. And then we'll follow on the Dan's talk, focus on neuroblastoma. So let's wait a couple more minutes to have more people slowly coming in. So yes, I guess we can start with the introductions. So as I said before, Yvonne is the Science and Technology Advisor for 10x, and she is currently based in Melbourne. So she joined 10x earlier this year. Is it earlier this year? Yeah. But she has a wealth of experience on genomics technology. She's been in a few companies, and then she has a lot of experience on that. And today she's going to share a little bit of the applications for 10x on the portfolio of 10x for solutions for oncology. Now, I would like to remind everybody that the questions will have questions at the end, both for Yvonne and Dan, and any technical questions that you might have, or questions about the facility at UTS, anything that you are, like any question, will have a bit of a time at the end of the seminar. But in the meantime, you can, please feel free to post your questions in the chat. It's not like as they go, so you don't forget once you have the question, and then we will be addressing those ones at the end of the seminar. So I think that's a bit of a 4 minutes past 10. So I think we can start with Yvonne's talk. Over to you, Yvonne. Thank you very much.
Right. Thanks, David. Thanks for the introduction, and good morning, everyone. Thanks for joining our third seminar this morning, and I want to take the first 20 minutes to give a quick introduction on some of the key technology platforms that 10x Genomics has, and some applications for cancer oncology in general. And I want to start today with this sort of timeline of innovations that 10x has released since its beginnings in 2015. And, you know, as David said, I joined in the beginning of this year, and it's actually been absolutely incredible, not only to learn what these already launched solutions do, but also witness since then some of the new launches that we've had, and how these technologies advance all these different fields. And specifically today, with the focus of oncology, the field is, you know, tremendously moving, utilizing some of these technologies here that you see. We can't really go into all of them, but I have picked a few of these solutions. So the single cell gene expression solution, the immune profiling solution, and specifically also our Visium spatial gene expression solution. And we'll just show you some examples of how they can be utilized. And what's actually really highlighting the single cell application and how it changes discovery and treatment for cancer was quite well highlighted in this publication here in Science in 2018, where they really focus on the different techniques and what they actually advance in the field of cancer, specifically tumor profiling using single cell technologies. And that's really seen as paving the way for many more discoveries that will come from these improved technologies. And obviously they are all, you know, they're not perfect, they all have some form of limitation, but I think where we're at right now, the advances that can be achieved, I think are quite tremendous, and some of those I'll highlight today as well. And to begin with, again, showcasing you the sort of the main technology platforms that 10x has. The third one here on the right, the in situ platform that we have, or are close to releasing from the acquisition of Redacor and Katana, is coming. But what we have right now is the single cell Chromium single cell solution, where you input single cell suspensions or nuclei suspensions from a variety of starting materials, whether it's tissue, frozen tissue, whether it's cell cultures, etc. And then we have 4 main assays that you can actually plug into this solution. And then we have our Visium spatial transcriptomic solution that is enabled for fresh frozen tissue sections, as well as FFPE tissue sections since recently for mouse and human. And that really just requires you to run this on the Visium glass slide. So there is no actual specific platform instrument required from 10x to undertake this. So looking at the Chromium single cell platform here, our Chromium Controller, the size of a shoebox, so very small. So if you do get a chance to go into the facility and have a look, it's really quite small and compact. Runs these 8-channel microfluidic chips that you can see here, and we can plug in these 4 different types of assays onto the chip as needed. And as I said, it's an end-to-end workflow. So you prepare the cell suspensions or nuclear suspensions, and that is, as Lou mentioned in our very first seminar series, you know, that is probably 85, 90, if not more % of the experiment that you put the effort in to generate these cell suspensions and nuclei suspensions. And then you load that onto the microfluidic chip, the encapsulation there into the nanoliter droplets with our 10x barcoded gel beads. Really, it only takes about 18 minutes, such a run. And then off the chip, you perform a series of barcoding and library preparation steps. And that leaves you with Illumina compatible sequencing ready libraries. So they all have each assay has sort of different recommendations for sequencing, and we can point those out specifically once you enter into one of these assays. But what I want to highlight with this end-to-end workflow solution here is that we have the analysis software and the visualization software. So that's Cell Ranger for single cell, Space Ranger for spatial, and Loop Browser available for downloading for free, or you can utilize third-party software tools as well. So you're not locked in there. So here is at high level what occurs on these microfluidic chips in our Chromium single cell instruments. So what's really the core to this GEM technology, gel beads in emulsion, where we input millions of these 10x barcoded gel beads, and we combine those with reagents needed, but of course with your sample, single cell or single nuclei, and encapsulate these in the partitioning, or surrounded by partitioning, or but encapsulating these in the nanoliter droplets. Some of the key specs for this instrument are that you can run up to 8 samples. So I mentioned there's 8 channels. If you don't multiplex, 10x also has the CellPlex kit available to multiplex up to 12 samples. But the dynamic range here that I want to mention starts from about 500 cells per channel up to 10,000 cells that you can capture in each individual lane. We have a multiplet rate of about 0.8% per 1,000 cells, so that's something to keep in mind, specifically when you start multiplexing in sort of subsequent experiment, as here, the more cells you use, the higher the multiplets are that you are generating. And then we have a very high capture rate of up to 65%, depending on cell type and nucleus type that you're using. So for cancer specifically, or for translational clinical research, this platform and the specific assays that you can plug in, not only are sort of replacing slowly some of these techniques that are currently being used, but they complement, I think, data that's already being generated. So you can, with the single cell gene expression solution, you can capture the whole transcriptome. With the immune profiling solution, you can look at T and B cell receptors in specific sequence and specific antigen interactions as well. With our feature barcoding technology, we enable shortly high throughput protein screening. And something that I want to go into a little bit today is the Crispr screening, Crispr perturbation screening that you can undertake with the 3' gene expression solution feature barcoding, as well as looking at chromatin structure with our ATAC-seq assay. So all those, really feed into some of these 4 main fields or questions that researchers are asking in terms of identifying tumor heterogeneity, identifying the tumor microenvironment, specifically looking into advancing immunotherapies by characterizing immune cell modulation and tumor progression. So in the field of immuno-oncology, and then therapeutic development, where with the single cell data, you are enabling, the discovery of new novel therapeutic targets, for example, and can explore new modulate cell populations as well with these, to just name a few here, pick a few from these. So the majority of the examples out of these, I picked where groups have utilized the gene expression solution here combined with the feature barcoding solution. So what that means at high level, I just want to explain here on this slide, is that we capture the 3' gene expression. So we capture the 3' end from the mRNA targets of the cell with our 3' gene expression gel beads. But that's not the only thing that you can capture of each individual cell. You can, on top of this, look at cell surface protein expression, utilizing antibodies, detecting the specific protein that you're interested in, and they have to be conjugated with a small oligo that can be captured in our biochemistry. So it will have to be complementary to one of our capture sequences on the gel bead. In addition to the cell surface protein, the 3' gene expression assay, as I mentioned, also enables CRISPR's perturbation screening. And how this works is that you would basically, in the design of the guide RNAs, incorporate the capture sequence 1 or capture sequence 2 in the stem or in the loop of the guide RNA. And then with that design, we are actually able to directly detect this in each perturbed cell at the end of your screen. So specific examples here, beginning with looking at the tumor microenvironment, excuse me, of a colorectal cancer sample. We are looking here at 8,370 cells that we have partitioned and analyzed using the 3' gene expression kit. And what you can see here is that a large number of cell clusters have been identified. These purple highlighted immune cells here have been annotated using the gene expression markers that are published in databases. So we have not added in any cell surface markers here, and specifically in this assay. We can also identify, several different tissue types, and of course, we can also identify the tumor cells in this data set. So what this also shows is that amongst the large number of immune cells is that there is, because of the relatively large cell number that we are looking at here, that we can actually distinguish between the different types of T cells within this particular sample. So that can be, a target for you to look at potentially what could be utilized for immunotherapy. We can also look at tissue cells versus tumor cells to look at or identify biomarkers and drug targets. And here what is also visible within the B cells is that we see a clear differentiation of these B cells into plasma cells, suggesting a robust B cell response in this tumor. So from those, just from the single 3' gene expression run here, you can conclude a large number of potential targets for therapy, for example, or for biomarker discovery. The next example here looks at the single-cell transcriptome network in gastric pre-malignant lesions and early gastric cancer. This here is a cell report from a group in China. They have utilized a 3' gene expression RNA sequencing here, and what we see here on the right is the expression profiles generated of a bit more than 32,000 cells, and they generated a cell atlas encompassing these pre-malignant and early malignant samples. So in the middle here, you can see the mucosa biopsies they have collected and the different stages and how many, and that's and then they have run this using the Chromium Controller. So in this large population here, they have identified very small groups of cells that they were able to analyze further in subsequent studies. That's not shown here. And they've commented, that some of those were not able to be identified, just utilizing bulk RNA sequencing experiments, which this group had conducted previously to running these single-cell transcriptome analysis. So they were able to go ahead and then identified specific genetic markers for potential clinical utility as diagnostic markers in this cancer type. This study here looked at, again, just simply using the 3' gene expression solution to screen triple-negative breast cancer patient-derived xenograft models to understand the high variability in responses to EGFR therapy. So here we are actually only looking at 3,500 cells from exceptional responder PDX from the exceptional responder PDX model. And in this marked highlighted black circle here, the authors identified subpopulations displaying distinct biological features, including a mesenchymal cluster which highly expressed EGFR that functioned as tumor initiating cells. And that suggested to them that they could investigate this further to determine if these EGFR associated tumorigenicity plays a role in the predicted treatment response for these cancer patients. Another application for the 3' gene expression solution is to look at underlying transcriptional mechanisms of immunotherapy resistance. And here in this example, the Hutchinson Cancer Research Center published this data here that you see looking at Merkel cell carcinoma patients. And they've run here single-cell RNA sequencing of tumor biopsies. In blue, you see biopsies collected pre-treatment, and then also biopsies collected after these patients had run into relapse. And you can see that there is a massive difference in expression between the different tumors. And the authors looked into this further to understand what exactly is happening. And on the right here, they've picked specifically, or we are looking specifically here at the expression of HLA-B. And what we can see here is that in the relapse a group of cells that is basically lost the expression, the HLA-B expression, which could explain why the immunotherapy resistance occurred. So the tumor had basically shut off transcriptionally the expression, and although the T cells could still recognize the antigen, it could no longer be presented. So this was of particular interest for the group because the transcriptional HLA repression is reversible. And so they're starting to look into, how this could be prolonging the immune therapy or the immune response in these particular patients, and showing how here single cell really sort of provided that insight to take this research further. Next, I want to touch on CRISPR specific with an example on cancer immune therapy. And here on the right, what you can see is just a graphic of these capture sequences that I mentioned that have to be incorporated into the guide RNA pool design. That you can then basically directly read with our 3' gene expression assay on the 3' gel beads. So the CRISPR for the 5' immune profiling solution is coming next year. And the advantage here will be that you could already use existing guide RNA pools, so you don't have to design new pools utilizing these capture sequences, as the capture, the target is going to work slightly differently. And utilizing CRISPR, what's the advantage of doing this CRISPR perturbation screening with the 10x solution? There is a few advantages here that are listed on the right, and one of them is that it makes the workflow, it shortens the workflow slightly, so it is faster than traditional screens, but it does depend on how quickly, I guess, your transduced cells are going to start expressing the guides, and then also how quickly will you be seeing these gene expression differences. But we have seen this time frame can be shortened down to just a few days compared to like week-long experiments previously. And then you do get a higher resolution with this type of analysis, and because you're detecting directly the guide that's expressed in each of the cells and measure the gene expression changes directly in each cell, you get a much more direct readout from a CRISPR screen. And this example here in cell highlights how the CRISPR screen in the standard Chromium controller environment can be used in screening primary immune function human T cells. And for this here specifically, the authors used a pool of 48 guide RNAs and transduced human T cells from two donors, and ran that through the Chromium controller. And what we can see here on the top is looking at the gene expression, the entire gene expression captured from all the T cells. And in the center, they have focused here on 4 specific genes that they were interested in and overlaid that. And then on the right, we can see the clustering by cell states, and that in total 13 different cell clusters they were able to identify 13 clusters in this screen here. And out of the pool of 48, the group was able to identify 7 guide RNAs that can perturb regulators involved in cell cycle, in T cell activation, other processes critical for immune response. And that's showcased here in the center at the bottom, these 7 guide RNAs that they showed directly in this publication. And this here highlights for this particular study how a fairly long genome-wide CRISPR screen could be shortened in this time, in this particular example here, down to 9 days, and that they then also performed some in vitro testing downstream to confirm some of the findings in this study. So from this CRISPR screen is just one example. I now want to switch gear and lastly give a couple of examples of the spatial analysis in cancer and why location matters with the Visium spatial gene expression solution. And here we are looking on the left on a tumor section that's been stained with H&E stain. And on the right, we are looking at tumor infiltrating lymphocytes. And in this particular instance here, we are looking at, are these lymphocytes successfully penetrating the tumor? And on the top, there's an example of a hot tumor where that has occurred, which relates to or means a good prognosis for this particular patient. And at the bottom in this example of a tumor section stained with H&E as well, on the right, the tumor infiltrating lymphocytes are not infiltrating, are being held back, which is representative of a cold tumor, which means poor prognosis for this particular patient. So here, it showcases quite nicely how the spatial context actually allows conclusions on prognosis, whereas a single cell expression profile would have just highlighted the presence of the lymphocytes rather than where the actual occurrence is. At high level, just wanted to showcase the workflow, specifically here for H&E and immunofluorescence. So you can detect protein on the same section, although only at this point was up to 6. We are limited to up to 6, and that's going to be extended soon. But you capture 4 tissue sections on one slide, you take an image, and then off the slide, you perform, again, barcoding and library construction steps to generate a sequencing-ready Illumina-compatible library. So these workflows can be completed in 1 or 2 days, respectively, depending on whether you're using FFPE or fresh frozen. And with this slide, I just want to highlight again, I'm sure you're all aware, but the resolution on our current Visium slides are about 5 to 10 cells. So we have each barcoded spot in the capture area with a diameter of 55 micrometers and center to center of about 100 micrometers. And with the high throughput, high definition HD solution that's coming, that will be reduced down to 5 micrometers per spot. So we'll bring this down to the single cell or subsingle cellular level. But of note also for the barcoded spots, we capturing polyadenylated RNA molecules with this solution. And last, I have one example of utilizing the spatial gene expression to study immune cell infiltration for HER2 positive breast cancer samples from the Cell Life Lab and what they've performed here. So if you're looking at one of these H&E stained section that the pathologist annotated, we can see, the tumor, invasive cancer cells, immune cells identified versus normal glands, fat tissue, etc. And then we are looking now at the gene expression heat map generated from the Visium solution, and that they have performed here on top. They sort of did a, that's the total immune score. So they've generated a scoring system to highlight sort of the intensity of expression that they detected. And then on the right here, they were looking at these specific immune cells that were identified based on their gene expression and where within the tissue the expression was highest. So you can see this with this distribution here. So when we look at the memory B cells in the heat map in the center, and then we look at on the right where the expression exactly occurs. And for the T cells, I think what the highlight here was, or the additional knowledge that they gained from the gene expression slide, was that the T upper 2 cells were expressed in immune cells not only, but also they were noticed in the fibrous tissue, in the invasive cancer, and in the tumor stroma, etc. So they were actually present in a lot more of the areas than it was annotated by the pathologist. And lastly, what you can do with this is you can also generate a 3D view of this, of these serial sections of tumors, and look at specific gene expression, the signatures of these tumors in these 3D models. So with that, I'll hand over to Dan. We'll take questions at the end, and thanks for your attention.
Okay. All right. I'm hoping everyone can see that. Thanks, Yvonne and David and the rest of 10X for putting this together and asking me to share some of our work. So today I'll talk a bit about our work in understanding drug resistance in a childhood cancer called neuroblastoma. So this is works unpublished and a bit of a work in progress, but it kind of proves some of the concepts in terms of the power of the single cell work and the utility of the 10X equipment. So just quickly, because Yvonne mostly covered this, but just to highlight again that cancer really is a multicellular disease. You have clearly lots of infiltrating normal cells within a tumor microenvironment that are very relevant to the prognosis and biology of the cancer. But even, of course, within the cancer cells, that you have a huge range of heterogeneity that is important, very important in terms of therapeutic response. So this heterogeneity in the cancer cells can have a genetic basis, of course, and that is either genetic aberrations or mutations that can drive kind of functional differences in cells. But there's also a non-genetic basis for tumor heterogeneity, which mostly we'll focus on today. But essentially, they both lead to a similar thing, which is the change in function of these cells, so that they firstly are cancer cells, but also important, as I've said, in terms of drug resistance and treatment failure and many other facets of cancer biology. And of course, single cell technologies like 10x are really beginning to understand this stuff in very high level detail. So since most of the talk will be about drug resistance, I thought I'd just start with a couple of the sort of key concepts of at least non-genetic drug resistance. Here, which is highlighted in this nice review paper, recommend reading, two concepts. One described as transcriptional priming, where a cell type has a unique advantage, allows it to survive treatment, in this case, the yellow cell, and then subsequently kind of expand to become a resistant cell type in that tumor. So that's a pretty simple concept of a clonal selection where one cell type survives effectively. So that's referred to as transcriptional priming. But probably a kind of more realistic idea of how cells are actually functioning is transcriptional adaptation. So this really comes down to the ability of cells to be plastic or change their phenotype at any given time. So in this figure, it kind of highlights that a cell or cell types can kind of change their function and adapt to and fro. And this is an escape mechanism. So you expose them in a particular environment like drugs or chemotherapy at high levels. They have the ability to change their phenotype, allow that short-term survival, and then ultimately resist and progress in the tumor. Where again, the cells can go to and fro between their phenotype depending on what's kind of most favorable to their survival. So this is a kind of problem when we're using single cell data to understand mechanisms. And so often we'll be faced with an issue with single cell data where we can't really determine whether it's a mechanism by transcriptional priming or transcriptional adaptation. So this is the kind of thing, or actually a much more simplified version of, but a kind of thing that we'd be faced with. A pre-treated on the left showing single cells and 5 cell types, and a post-treated sample that kind of enriches this one cell type as an example. So if we show that schematically, you can kind of see on the right that clearly the blue cell is the surviving cell, right? So this is an example of course, transcriptional priming. One cell cell type has an advantage, allows survival and tolerance against the drug action, and then ultimately expansion. But consider a kind of real world scenario where actually you're getting multiple mechanisms underlying drug resistance, where sure, you may have transcriptional priming, you've got the one strong cell, but it's also, of course, possible that cell types can change and adapt by plasticity mechanisms. So in this case, you may have, for instance, the yellow and the green cell type having adaptive capacity to kind of generate this post-treated resistant cell type. So, of course, given normal single cell data, we really can't determine how these are occurring, whether it's a priming, adaptation, or a combination of both mechanisms. So that's where lineage tracing comes in. So the principle of lineage tracing is really just to mark the individual cells before treatment here. So if we add a kind of mark or tag, in this instance, we're just referring to it as the A tag, we can actually trace then that A expanded in response to treatment. Whereas C, it may not have expanded, but it's actually then changed its cell type. So we can determine then that mechanism is by adaptation, and similarly for lineage D here. So that's kind of example of why we want to use lineage tracing for drug resistance studies. So the actual method we used was developed by Samantha Morris called cell tagging. And the principle of cell tagging is that in the 3' UTR of GFP, you have this 8 base pair randomer, or what she refers to as the cell tag. So a library of these GFP molecules is transduced into cells, and the combination of these barcodes in cells then can kind of mark their lineage because they're heritable. Once the cell receives the cell tag, divides and expands, you're kind of creating these lineages based on these unique combinations of these actual cell tags. And I guess the nice thing about this technology that extends upon kind of more of the older versions of lineage tracing is this is from an expressed molecule. So GFP is expressed, it's in the mRNA molecule, we can detect it using single cell RNA sequencing. So we get, of course, the lineage, which is our objective, but at the same time, we get whole transcriptome of that same cell. So we understand the whole transcriptome of the cell, but at the same time, the interconnections of the cell lineages. And Sam very nicely demonstrated this technology in cell reprogramming in her paper a couple of years ago, where she was not for cancer, but for reprogramming, cell reprogramming. She used this to track the reprogramming of certain lineages from a fibroblast to what she calls an induced endodermal progenitor. And she highlighted 2 major trajectories, one which was kind of inefficient and one which was efficient, and subsequently used that information to the genetic features of the efficient reprogrammer to improve reprogramming. So the actual application of that was really nice as well. But coming back to neuroblastoma, I won't go into the biology too much, but it's a kid's cancer, as I said, of the peripheral nerves, and it kind of a range of responses in patients, and it's becoming quite clear that the patients that respond poorly have heterogeneous disease. So we and others have started looking at this using single cell work, and effectively, to summarize quite a lot of work, we described 3 main cell types: an adrenergic cell type, a mesenchymal cell type, and what we refer to as a transitional cell type. So, but I guess the major outcome of our work and others is that we really don't know the interconnections between these cells. So it's theorized that they transdifferentiate between these cell states, and cell plasticity is important. So we wanted to do this with cell tagging, so we needed a cell system for that. Fortunately, there's a pretty well-known neuroblastoma cell line called SKNSH. And in this case, you can see here, this is the single cell RNA data showing high expression of each one of those cell type signatures. So we got the adrenergic signature more highly expressed here, transitional signature more highly expressed here, and mesenchymal. So they're pretty exclusive of each other, these signatures. So supporting the idea that the kind of a continuum of expression for cells adapting, and supporting kind of the idea that is a good cell system is that the clustering kind of conform nicely to these adrenergic, transitional, and mesenchymal cell types, all within one cell line. So to depict this a little bit cleaner, and I'll be using a lot of these in the talk, we've used these ternary plots. So effectively, all we're doing is summarizing each cell according to the expression of the 3 signatures. So this is a 3-coordinate system where cells can exist anywhere between adrenergic and mesenchymal cell types, or in between towards the kind of transitional cell type. So the coloring here just indicating where they fit in that spectrum. So we cell tagged this SKNSH cell line, and then what we had to do first, though, was we transduced the cells with the cell tag library, subcloned only 250 of the cells because we didn't want too many lineages. It would have been too hard to track with the single cell RNA sequencing. We expanded those 250 cells into a maximum of 250 lineages within that culture, and then used the 10x to do the single cell RNA sequencing. So this is some of the data just to show that we were able to detect cell tags And on the left here is just a correlation plot between cell tag expression profiles, where if they had high correlations, like these dark blue boxes here, we were able to call them in that they formed a clone or a lineage, and the color just indicating the different clones that we were able to call from that. And you can see there's the kind of wide range of sizes, and that's highlighted here, that of our sort of top 50, I think there's 56 clones. We had ranges of 400 cells right down to 10 cells per clone, and this kind of arises because of the sort of variable proliferation rates of cells once we expand them. But still, these were the high confidence clones. We actually got quite a lot more, but we prefer to stick to those which we're very confident that they actually do form a separate clone. And on the right here, we can see the distribution, and not that I really want you to see anything in particular here, other than that the clones are kind of pretty randomly distributed, or at least diversely distributed, meaning we could already tell here that within individual clones there was a spread of transcriptome, suggesting the cells spontaneously adapt their phenotype. But our main purpose was to understand drug resistance mechanisms. So we undertook this pretty simple experiment where we treated these SK and SH cells with the DMSO vehicle or chemotherapy, very well-known chemotherapy Cis Platinum, which is used in neuroblastoma, but of course many other cancers. DMSO just acts as the control here in the vehicle. We wanted to include this to control for the natural proliferation of the cells. But we took these 5 samples and did the 10x based single cell RNA sequencing on that. So to step you through the data in terms of how the transcriptome looks, we use the ternary plots as I described before. So this just shows the untreated sample and 24 hours of DMSO or 120 hours of DMSO. And thankfully, the actual vehicle didn't change the transcriptome terribly much at all. So that was good that the control wasn't really causing any aberrant effects. But with cisplatin, it was really quite an obvious shift, which was great for our purposes. And at least at the 5-day mark, you could really see a pretty strong shift towards the mesenchymal pole, where they sort of started over here as a mixture between adrenergic or transitional cells and maybe slightly mesenchymal, and really ended up very mesenchymal in nature. And to summarize, at least at the sample level, what's going on, again, each one of these dots corresponds to just the average of the individual sample. You can't see terribly much difference with the DMSO, but you get this really obvious shift or trajectory towards the mesenchymal pole with treatment. But of course, the cell tags allow us to pull out the individual lineages, which was our main purpose here. And this then also shows us our ability to quantify the proportion of each sample that is occupied by those lineages or clones. And so here's kind of complicated plot, but effectively, if you focus on the cisplatin treated samples here, we see these subclones in purple, or lineages in purple, which obviously are expanding. They're becoming much more prominent feature of the whole population after cisplatin treatment. And on the flip side, you get these cisplatin depleted clones as well. So there seems to be at least two types of clones in this case, where one which is naturally enriched or kind of tolerant or resistant to cisplatin, and those which are depleted or sensitive to cisplatin. So to step you through the lineage analysis, or the transcriptomic change of individual lineages or subclones, I'll just base it on increasing levels of complexity. So again, just showing you the sample level trajectories as described before, we get this cisplatin shift. Now this is comparing the cisplatin depleted versus cisplatin enriched clones, and it's kind of interesting that both clone types want to shift towards the mesenchymal, and so there's clearly a cisplatin is driving mesenchymal changes in all of these cells. But the difference was the kind of starting point. So those clones which were enriched or resistant to cisplatin kind of started more to the top of this triangle or ternary plot. And whilst only occupying a minority of the population to begin with, ended up expanding to become a majority. And so it was really a stark difference in terms of their change in proportion, but not a huge difference in terms of their trajectory. They just sort of started at a slightly different point, the enriched versus depleted. But of course, we were really interested in the individual clones. And so this is a bit of a mess and not really expecting you to see anything much more than what I described on this section, but we can see all the individual trajectories that the clones can take can be really quite diverse. So that was our best attempt at summarizing it, but it's still interesting to note that some clones really do have kind of strange trajectories in response to cisplatin, really to us at least highlighted how diverse or variable responses of cells can be to chemotherapy. So again, just highlighting probably the key difference we could see though was the starting point, or another way of saying that is that the cells were transcriptionally primed, meaning they had pre-existing characteristics, or these enriched clones had pre-existing characteristics that allowed cisplatin resistance. So just quickly to look at this quantitatively, we described them in terms of a transcriptional priming score where they can fit within this kind of spectrum from adrenergic to mesenchymal, and then looked at that in relation to their relative cisplatin enrichment. And you can see it was a pretty nice correlation. So it was really quite predictive. Where a clone falls in terms of this transcriptional priming score is strongly predictive of whether it's going to be enriched or depleted of by cisplatin. And of course, what are the genes? And we could look at this using our parallel transcriptomic data. And we compared cisplatin enriched clones versus depleted clones, and could see features of sort of mesenchymal stem cells, I guess you could say, or neural progenitors, and some interesting genes of note for cancer reaches EGFR, and some of these stemness factors or progenitor factors. ACL1 is well known in neuroblastoma development, sympathoadrenal development. But then you also have these genes featuring kind of sympathetic neurons or differentiated phenotypes in the depleted clones. And indeed, where we looked at the pathway analysis, we were able to see that the main feature that predicted cisplatin enrichment was really a stop in differentiation. So negative regulation of neuron differentiation here, etc., contrasting to the depleted clones, which is promoting differentiation or features of neuronal kind of cells here. So just finally, just to summarize, that this is just all work in one cell line, and of course we want to extend this and make it more generalized, but this is what we've done so far to learn whether this is just a more general feature in terms of drug resistance. We created a gene signature from those genes that I mentioned in the last slide, and explored that gene signature in the genomics and drug sensitivity database, which has screened 23 neuroblastoma cell lines by 179 drugs. And down the bottom here, you can see that the IC50 of those individual cell lines to cisplatin is positively correlated with the cisplatin resistance signature. So these genes seem to predict the resistance in many cell lines, at least by this bulk data that we have here. And kind of interestingly also that other chemotherapies that are used in neuroblastoma also have this positive correlation. So these genes, we would hypothesize at least, are predicting general resistance to chemotherapy. So with that, just a quick summary, we saw two cell types really exhibited in this cell line. The sympathetic neuron or chromaffin cells, which is the major population, is primed for drug sensitivity and was plastic, but not very efficiently. The sympathetic progenitor, which is a minor population before treatment, but it is primed for drug resistance, has efficient plasticity, really converted to the mesenchymal pole quite well. And then when we add cisplatin, we really all cells are adapting. So it's not just like a simple concept of only one strong cell type. We're kind of getting priming plus adaptation. Really, these mesenchymal derivatives have features of EMT maybe, or potentially dedifferentiation. So I guess a highlight of the work was that transcriptional priming and adaptation are not mutually exclusive. We would argue in this case that our cells are primed to adapt better, put it that way, as a survival mechanism, these sympathetic progenitors. We're interested in looking at drugs, of course, as a potentially targeted agent to overcome this, and then interested whether this is just a short-term effect, or whether if we remove drugs, would they therefore revert phenotype. There are lots of questions around how this relates to actually a patient tumor. So with that, I'd just like to thank my contributors, in particular the team at CCI who undertook all the lab work here, my colleagues here at UTS, and Sam Morris in Washington University for the development of the cell tags, and the funding bodies. And thanks very much for listening. We can't hear you, David. Yeah, I'm not sure if it's just me, but I can't hear you, David. So.
Me neither, David.
I can't hear you either.
Well, I guess maybe we just open the floor to questions then, right? If there's anyone from the audience or on the panel, any questions? Still can't hear you, David.
There's no questions in the chat.
I could start with a question for you, Yvonne, if you like.
All right.
Yeah, I was kind of interested in the Crispr work, and I mean, I see the advantages in terms of scale and a unique kind of output, and we're particularly interested in this. But one of our objectives is to look at combinations of Crispr perturbations in the same cell, so which I see is possible using this technology, but maybe an issue in terms of numbers, like in terms of how many cells you would have to look at and how few guide RNAs you'd have to look at. But do you have any comments on kind of the feasibility of those sorts of experiments?
Yeah, it's a good question. So with I guess with a 10,000 per lane, you are a bit limited, right, in the number of guides you can look at. We also, recommend a sort of a minimum number of cells per guide as well to actually make meaningful or generate meaningful data. But if you are interested in larger numbers of guides, I think the Chromium X with a higher throughput might actually be a better option for that. So it's, I guess, specifically designed to run more cells, enabling, larger throughput CRISPR screens. So I don't know what you had in mind. I think what the paper that I showed there with 48 guides, that's probably around the number that was feasible with a standard Chromium Controller run. anything higher than that, you know, either you run several chips or you combine it on a high throughput screen on the Chromium X.
I'm sorry, my internet just cut out in the middle of that answer there. But yeah, I was understanding that mostly what you were saying was that you really just need to increase throughput to account for that kind of random association of multiple guides per cell, right?
Yes, and you know, as the Chromium X with the high throughput reagents, where you basically screen like twice as many cells per lane, would make that a bit easier, right? Rather than, I guess, running several chips on a Chromium controller. But yeah, basically that's what you'd need, higher numbers. And I was asking, I don't know if you've cut out then or got back in, but the publication that I showed where they had about 48 guides that they used, that's sort of the number. I think the highest, I think we've actually seen 80 with the Chromium controller, but that's probably the top end of guides that you can use.
Yeah, I see. Thank you.
Can you hear me now?
Yay!
Oh, sorry for
you.
that. I'm back. Sorry, I couldn't introduce you, Dan, properly because I think my microphone was off in the middle. So anyway,
no worries. All good.
So well, I'm aware of the time, but so thank you both for being here today and for your talk. Dan, I think that you're showing very interesting data. I actually have a question for you. Like if everybody wants to stay a few seconds more, I'd like to ask you: have you tried, you know, like the ternary plots that you're showing to infer trajectories based on your signatures? But have you tried to do an unbiased trajectory analysis or unsupervised trajectory analysis to look at whether, you know, there's multiple fates within those ones, because sort of you're defining those three states that you were showing based on previous biologies? That is there anything else that you can predict that is not being shown before? Perhaps, you know, multiple mesenchymal subtypes or subclasses.
Yeah, well, I mean, we've looked at trajectories in terms of, say, a UMAP, and in terms of like that ternary plot is really just a compromise of us trying to simplify the biology enough to show trajectories that are meaningful. But you're right that taking an unbiased approach, I mean, we effectively see the same thing, the trajectories in terms of the shift towards the mesenchymal arm now, but the actual exact pathway that is undertaken, the best we could simplify it was those that are enriched versus those that were depleted in terms of the subclones. But I think really, if we look at individual clones, we see quite diverse patterns, and so I'm not sure we could easily say it's a different mesenchymal subtype or not, because one of the things that's hard to highlight in the data that all of the kind of average or the centroids that we show for the clones are made up of, you know, up 10 to hundreds of cells, and those cells also have spread or plasticity. So even within a single, you know, sample, there's spread, and it definitely has discrete patterns, but you wouldn't say necessarily that they're exactly all falling where the centroid is. That's just our best way of summarizing the trajectory. So I think there's a lot to that, in the sense of drug escape mechanisms that really cells can react in pretty you know, weird and wonderful ways to any environmental exposure. And for cancer, that kind of means cells find a way to survive. And but it'd be interesting to see where we can use this information to actually help us choose better therapies, you know, at least to even just illustrate the mechanism that we can either prime cells to be sensitive to other drugs, or just use that known heterogeneity to choose combinations of drugs or something like that. So yeah,
that sounds like a very good way to understand that. So it's great. to see this
So
technology
Daniel,
combined
and
with
on
the
that,
techniques.
on the note of the trajectory, so when you do your analysis, what makes you select the trajectory algorithm over a velocity? So the trajectory will not tell you what direction, it will just tell you, the path, but it will not tell you what direction it's going, right? So where the velocity will allow you that. So, and the underlying idea is the same, but one you have direction and the other is just kind of, the biology, you put the direction.
Yeah. So even if we looked at the probably easiest way to look at that particular problem is just look at the untreated sample by itself, and because then there's no strong environmental exposure like the drugs, which are driving change anyway. And when we did that, you kind of show a RNA velocity trajectory that has a root in those transitional cells. So they have the capacity to differentiate down either pole and in terms of directionality. And so we see, I'm calling them transitional cells, probably not the most accurate name. In fact, we're just calling them probably more like progenitor cells. And that they're the kind of root of differentiation and which spontaneously goes down towards the either pole. But even with therapy, of course, that's really driving a push towards the mesenchymal pole. It shows that the trajectory can maybe differ to that base or steady state trajectory. So my feeling is that the cells can move in many directions, and it really, I think, we'll try and learn about directionality in response to the drugs. Also, using RNA velocity is probably a good idea and things like that. But we can at least know that that's time point data. We know time 0 versus time 5, that the trajectory therefore is in that direction. Yeah. So.
Right.
Thanks.
Well, thank you very much. I'm cautious of the time, and I would like to thank the speakers Yvonne and Daniel for the insights on this cancer biology and translational oncology, and also Lou for joining us, and Karen for organizing all the logistics around the meetings and the materials for promotion. So thank you very much, everybody. And I'll see you in 2 weeks' time. Thank you.
Thanks, David.
Thank you.
Cheers. Bye, guys.
Bye.